Hello students, this is your Prasanna ma'am from NEET World. Today we are here to discuss the topic Monera from biological classification. If you look into the general characteristics of Monera, Monera includes all the prokaryotic organisms and bacteria are the sole members of the kingdom Monera. Bacteria are the sole members of kingdom Monera. And if you look into a prokaryotic cell, no nuclear membrane is present, no nucleolus, no chromatin, no histone proteins, and there are no membrane bound organelles. Other than histone proteins, what do you have ma? Some positively charged polyamines. Some positively charged polyamines. These positively charged polyamines will bind along with the negatively charged DNA and they will place the DNA at one position in the cytoplasm. Even though the DNA is not membrane bound, their position is at a particular site in the cytoplasm. That is because the DNA is connected with some positively charged polyamines, wherein DNA is located at a particular position in the cytoplasm, which is called as nucleoid. This is called as what ma? Nucleoid. And there are no membrane bound organelles in a prokaryotic cell. If you look into any prokaryotic cell, be it eubacteria, archaebacteria, mycoplasma and actinomycetes, there is a rigid cell wall present. Rigid cell wall. In eubacteria, peptidoglycan is the cell wall. What is the cell wall in eubacteria? Peptidoglycan. Archaebacteria cell wall is constituted with some polysaccharides and sugars. Mycoplasma is the only Monera, only bacteria in the Monera which doesn't have any cell wall. It doesn't have any cell wall, and this is what ma? This is the smallest cell. Smallest cell is mycoplasma. Next, actinomycetes. This type of bacteria will have mycolic acid in its cell wall. Next, nucleoid or incipient nucleus is composed of double-stranded circular DNA, RNA and polyamines which are positively charged Next, if you see ribosomes. Ribosomes in a prokaryotic cell are located at two sites. One is in the cytoplasm where you will call it as cytosolic ribosomes, and other is attached to the cell membrane. The ribosomes which are attached to the cell membrane are involved in protein synthesis which are translocated out of the cell. Okay, the ribosomes which are attached to the cell membrane are involved in the protein synthesis for translocation. The cytosolic ribosomes which are present in the cytoplasm, their protein synthesis is involved in the metabolic activities conducted by the cell. Okay, and if you keenly look into a bacterial cell, it is first uncovered with a cell envelope, then a cell wall, then plasma membrane. Okay, so if you see, what is the nucleoid constituted with? Double-stranded circular DNA, RNA and polyamines. And what beta? Polyamines. Next, ribosomes are of what type? 70S ribosomes, wherein larger subunit is 50S, smaller subunit is 30S. Next, high metabolic diversity is seen in the kingdom Monera. High diversity with respect to morphology and habitat is seen in the kingdom Fungi. Metabolic diversity, though your answer should be the kingdom Monera. So, what is the metabolic diversity that you see? In nutrition, you have two types of nutrition conducted by the organisms of Monera. One is autotrophic and the other is heterotrophic. In autotrophic also photosynthetic autotrophs, chemosynthetic autotrophs. Heterotrophic, there are three modes of nutrition under heterotrophic. Those are saprophytic, symbiotic and parasitic. Next, if you see respiration. Respiration also is of two types in the kingdom Monera. What are they? Aerobic respiration and anaerobic respiration. Under aerobic respiration, you again have two types of respiration. One is obligate aerobic respiration. Another type are facultative anaerobes. What does this term mean? Facultative anaerobes. Facultative aerobes. These are the organisms which maximum they will conduct aerobic respiration only. But then whenever those organisms are deprived of oxygen, when there is no availability of oxygen, at that point of time they will conduct anaerobic respiration also. They will conduct anaerobic respiration also. Which means even if the oxygen presence is not there, even if there is absence of oxygen also, they will conduct the respiration. That is called as facultative anaerobe. Obligate aerobes means compulsory, 100% they need oxygen to conduct the respiration. Next, anaerobic respiration. Obligate anaerobes, they doesn't need any oxygen. Complete anoxygenic conditions are required to conduct the respiration. Then it is called as obligate anaerobic respiration. Facultative aerobes means most of the time they are conducting anaerobic respiration. Only at some points, whenever there is availability of oxygen, then only they will conduct oxygenic respiration. Those are what ma? Facultative aerobes. Next, reproduction. Reproduction is of two types, asexual and sexual recombination we will call it as. Not sexual reproduction because there is no gamete fusion or gamete formation, just the gene recombination takes place over here. That is why we will call either gene recombination or sexual recombination, which is of three types. One is conjugation, transformation and transduction. Next, if you see bacterial appendages. Over the cell surface, two types of appendages will arise from a bacterial cell. One is motile and the other is non-motile appendages. Motile appendages are flagella, which includes flagellin protein. What does flagella? Made up of flagellin protein. Because they are prokaryotic cell. If they are eukaryotic cell, then it is tubulin protein. Flagellin protein. What is the type of arrangement of microtubules over here ma? It is 9 plus 2. Okay, it is what type of arrangement? 9 plus 2 arrangement because it is flagella. Next, non-motile type are pili and fimbriae. They will just be helpful for the attachment of bacteria over the substratum or onto the host cell. Next, classification if you observe, based on the presence or absence of the cell wall and composition of the cell wall and habitat. Based on the presence or absence of cell wall, habitat and the composition of cell wall, four types of bacteria are classified. They are archaebacteria, eubacteria, mycoplasma and actinomycetes. Archaebacteria, eubacteria, mycoplasma, till here it is enough for the NEET aspirants. Actinomycetes also have to be taken care of. You need to look into actinomycetes if you are preparing for EAP set. Next, if you look into archaebacteria, these are the most primitive monerans. You can call it as ancestors. Okay, they are ancestors. First arised organisms in the Monera are archaebacteria. That is why they are called as primitive monerans. They are called it as what ma? Primitive monerans. They occur in the extreme harsh habitats. This is because of their cell wall composition. They can occur in the extreme habitats. This is because of what ma? Because of their cell wall composition. The cell wall of archaebacteria is composed of polysaccharides and lipids. Okay, polysaccharides and lipids which are unsaturated. Okay, you can call it as monolayered lipids. Monolayered. Next, based on the type of habitat where the archaebacteria is living, we again have three types of archaebacteria. They are halophiles, thermoacidophiles and methanogens. What are they beta? Halophiles, thermoacidophiles and methanogens. Halophiles are those archaebacteria which live in extreme salty conditions. Thermoacidophiles, those bacteria which live in hot springs and they tolerate high temperature, high acidic environments and they can survive in low pH. Methanogens, the bacteria which grow in marshy areas and they will live in the gut of ruminants. Okay. and they are helpful in the synthesis of biogas. Examples of halophiles, examples are very important beta, you need to fix it in your mind. Halophiles examples you can easily remember halobacteria and halococcus. Thermoacidophiles examples include thermoplasma and sulfolobus. Methanogens examples methanococcus, methanobacteria. We have already discussed methanogens can survive in what ma? They exist in gut of ruminants, wherein they will convert carbon dioxide, methanol and formic acid into methane. This methane from cow dung, can be utilized for production of biogas from dung of the animals. This methane can be utilized for what? Production of biogas, which is very, very important, need to be remembered. Okay. Next, if you see here it is eubacteria. Eubacteria are cosmopolitan in distribution, and if you look into the cell of a eubacteria, of course, cell envelope here is composed of three components. One is glycocalyx, second is cell wall, and third is cell membrane or plasma membrane. Cell membrane or what beta? Plasma membrane. Glycocalyx can exist in two forms, one is capsule and other is slime layer. Here it is capsule, which is thick and tough. Below the capsule you have cell wall, which is composed of peptidoglycan or murin, okay, peptidoglycan and murin. Mesosomes, these are plasma membrane extensions into the cytoplasm. Mesosomes functions include DNA replication, cell wall formation, respiration and secretion. And ribosomes, which are present in the cytoplasm, are playing crucial role in what ma? Protein synthesis, and those ribosomes which are attached to the plasma membrane, their function is to synthesize the proteins which are needed to translocation, okay, which are needed for translocation. Next, based on the shape, eubacteria are grouped to 4 categories based on their shape. If they are circular or spherical, you will call it as cocci. In group though it is cocci. If they are singular, then it is coccus. Okay, rod shaped bacilli. Rod shaped what ma? Bacilli. If it is singular, you will call it as what beta? Bacillus. Next, spiral shaped. Okay, spiral shaped spirilla, or you can call it as spirillum if it is singular, spirillum. Vibrio, these are what? Comma shaped vibrium, you can also call it vibrio if they are in group. If it is singular, so you need to call it as vibrium. Next, of all these 4 categories of eubacteria, the most common bacteria is Bacillus, and this is the most pathogenic bacteria as well. Next, metabolic diversity. If you see, you have 2 types of metabolic diversity. One is anabolism and the other is catabolism. One is what ma? Anabolism, where it includes the synthesis of complex molecules like carbohydrates, proteins, lipids, okay, which includes nutrition. Catabolism, breaking down of the complex macromolecules like carbohydrates, proteins and lipids into simpler molecules by releasing the energy whichever is required for the metabolic activities, that is what ma? Respiration. So, anabolism includes nutrition, catabolism includes respiration. Wide diverse varieties of nutrition and different pathways of respiration are conducted by different organisms which are included in Monera. We are going to learn it now. Okay. If you take nutrition, 2 types of nutrition, one it is autotrophic and the other is heterotrophic. Autotrophic nutrition, 2 types again in this, photoautotrophic and chemoautotrophic. Photoautotrophic, wherein it receives the sunlight, okay, sunlight or light as energy to synthesize the food material, whereas chemoautotrophs will take inorganic molecules which are around the bacteria to synthesize the food material. Heterotrophs, so three types: saprophytic, symbiotic and parasitic. Now if you see nutrition, that is photoautotrophic and chemoautotrophic. Energy used by photoautotrophs is sunlight, as we have discussed what it is ma, sunlight, and electron donor is of two types based on the requirement or based on the electron donor. This photoautotrophic bacteria are of two types. One is oxygenic they will conduct two types of photosynthesis. One is oxygenic photosynthesis and the other is anoxygenic photosynthesis. If it is oxygenic photosynthesis, then the electron must and should be water, like in cyanobacteria. Example of this is what ma? Cyanobacteria or BGA, where it conducts oxygenic photosynthesis. H2S is the electron donor for those organisms which are conducting anoxygenic photosynthesis. For example, chloronema and chlorobium. Okay, this is about photoautotrophic nutrition. Coming to chemoautotrophic nutrition, the energy used is ATP and this is from inorganic molecules. Here inorganic molecules will serve as electron donor. Inorganic molecules like nitrogen, phosphorus, ferrous and sulfur. Okay. For example, nitrifying bacteria is one of the example for chemoautotrophic nutrition. What are the types of nitrifying bacteria which are there? Ammonia, whichever is present in the soil, this ammonia is firstly converted to nitrate. Ammonia is converted to what ma? Nitrate by the bacteria Nitrosomonas and Nitrosococcus. Here the energy is taken from what? Nitrates, okay, nitrogen. Then this nitrate is converted to nitrite. Nitrate is converted to nitrite by Nitrobacter and Nitrocystis. This is also taking the inorganic molecules, that is nitrate. Here the energy is being taken from ammonia. Okay. So as such, as they are using the inorganic molecules, they play a very crucial role in recycling the nutrients. Very important question asked multiple times. Which bacteria are helpful in recycling the nutrients? Then your answer should be chemoautotrophic bacteria. Why chemoautotrophic bacteria are helpful in recycling nutrients? Because they are taking inorganic molecules as electron donor and ATP from them is utilized as energy to conduct the process of photosynthesis. Clear beta? Next, heterotrophic bacteria. If you look into here, you have three modes of nutrition under the heterotrophic mode of nutrition. One is saprophytic. Saprophytic are the majority of heterotrophs, bacteria whichever is relaying in the heterotrophic nutrition. Most of them are what? Saprophytic, wherein they secrete the digestive enzymes outside. The bacteria will secrete the digestive enzymes outside. This digestive enzymes will clear the complex bonds whichever are present in this organic matter. After simplification, the bacteria will ingest those simple molecules. Complex insoluble substance is turned to simple soluble compounds and they will take the simple soluble compounds. Symbiotic bacteria, for example, like Rhizobium leguminosarum, which is in the symbiotic association with leguminous plants, that is of the family Fabaceae. That is of the family Fabaceae. Next, few are parasitic, for example, E. coli and Salmonella typhimurium. Next, respiration. If you see, we have two types of respiration which are conducted by monerans. One is aerobic bacteria and the other is anaerobic bacteria. Aerobic bacteria, you have obligate aerobes and facultative anaerobes, which means most of the time they are aerobic. Whenever it is deprived of oxygen, whenever oxygen is not available, then they are acting as anaerobic. They can survive even in the absence of oxygen. they can survive in absence of oxygen. Sometimes, okay, facultative, you include the synonym sometimes. Sometimes they act as anaerobes, okay. Most of the time they are obligate aerobes only. Example for obligate aerobe is Bacillus subtilis. Example for facultative anaerobe is Pseudomonas. Coming to anaerobic bacteria, obligate anaerobes, it is obligate anaerobes and facultative aerobes. Obligate anaerobes means 100% it require anaerobic conditions only, anoxygenic conditions only to conduct the respiration. For example, Clostridium. Facultative aerobes, most of the time it is anaerobic, but it can survive even in the presence of oxygen. It can survive even in the presence of oxygen. If you take for example Chlorobium, example of facultative aerobe is what ma? Chlorobium, wherein most of the photosynthetic bacteria are facultative aerobes only. Again, important question, most of the photosynthetic bacteria are what type ma? Facultative aerobes. Okay, clear? Next. Coming to eubacteria example, that is cyanobacteria. This structure is very much important. This is given in your NCERT diagram also and EAP set. Okay. So, this diagram is very important. Look into it keenly. Two types of cells are present in a cyanobacteria. One is vegetative cell and the other is heterocyst. One is vegetative cell and the other is what ma? Heterocyst. Vegetative cell, if you see, inside a vegetative cell what components are present? Photosystem 1 and photosystem 2. Chlorophyll A is the pigment which is present in a vegetative cell, and the electron donor which it is taking to conduct the photosynthesis is water. When the electron donor is water, what type of photosynthesis it is conducting ma? Oxygenic photosynthesis. Heterocyst have a thick cell wall outside, it has thick membrane outside. This is impermeable to water, this is impermeable to oxygen. The membrane is what ma? Impermeable to oxygen. Why heterocyst membrane is impermeable to oxygen? It is because heterocyst main function is nitrogen fixation. What is the main function of heterocyst? Nitrogen fixation. And heterocyst has only PS1, photosystem 1. This generates ATP. That ATP is utilized for nitrogen fixation. For nitrogen fixation, 100% anoxygenic conditions are required. 100% what condition is required beta? Anoxygenic condition, because nitrogenase is the enzyme which is helpful for nitrogen fixation, and this nitrogenase is inactive in the presence of oxygen. This is inactive in the presence of oxygen. This is active in the anoxygenic conditions. That is why heterocyst membrane is very tough, which is not permeable to oxygen. Clear? Keep it in your mind. Next, the membranous extensions of plasma membrane towards the cytoplasm here are chromatophores. What are the membranous extensions here called as beta? Chromatophores. These contain pigments and this will receive the sunlight for conducting photosynthesis. Next, in the center you have centroplasm, towards periphery you have chromoplasm. Apart from these two, vegetative cell and heterocyst, you also have akinates. What are these akinates? These are the dead cells which are hard, tough and dead. They are helpful for reproduction. They are helpful for what beta? Reproduction. Okay, so this is the filamentous structure of cyanobacteria explained very keenly. Take a screenshot of this if possible. This is very important. Next. So what are they? They are nothing but they can exist in the form of unicellular, colonial, mostly filamentous, few are aquatic and few are terrestrial algae. They are also called as algae because they can conduct photosynthesis. They can prepare their own food material, that is why they are called as blue green algae. Again they are included in Monera because they have what type of cell? Prokaryotic cell without any membranes. They don't have membrane bound organelles, they don't have membrane bound nucleus, that is why it is prokaryotic cell. And as it is conducting the photosynthesis, preparing its own food, it is also called as algae, that is blue green algae. Now photosynthetic autotrophs, these are what? Photosynthetic autotrophs, and they are most primitive. They are most primitive type of photosynthetic autotrophs which are conducting what type of photosynthesis? Oxygenic photosynthesis. What type of photosynthesis is conducted by BGA? Definitely asked. Oxygenic photosynthesis. They form algal blooms in polluted water bodies. So they are good pollution indicators also. They are good pollution indicators. Forming algal blooms in the polluted water bodies is called as what, Nana? Eutrophication. It is called as what, beta? Eutrophication. Next, heterocysts, just now we have discussed. These are what, beta? Specialized cells to fix the atmospheric nitrogen. And the examples of cyanobacteria include nostoc, Amoeba, oscillatoria, okay, Alocera. All these are the examples of cyanobacteria. Next, achinates, we discussed, right? They are thick walled dormant cells in BGA. They germinate into new fragment once they are divided from the filamentous BGA, filamentous cyanobacteria. Other type of bacteria, if you see, they are Trichodesmium erythraeum, which is a marine cyanobacteria. What is this? Marine cyanobacteria. They give red color to the Red Sea. They give what, red color to the Red Sea. As they proliferate, they release some enzymes which make the sea appear red. These are what? Red cyanobacteria. Example is Trichodesmium erythraeum. Don't get confused between Trichodesmium and Goniolax, which is the example of dinoflagellates under Protista. Under Protista, you have one more example, Goniolax, which is releasing saxitoxin during their proliferation, which makes the sea appear red. And here also there is one more example under cyanobacteria, that is Trichodesmium, which lives in the Red Sea, that makes the sea appear red. As they proliferate, they make the sea appear red. Next, reproduction. If you see, two types of reproduction are there. One is asexual and sexual. Asexual reproduction, during favorable conditions, they conduct binary fission. During favorable condition, what type of reproduction happens under asexual? Binary fission. During unfavorable conditions, they form endospores. Sexual recombination or gene recombination includes three types. One is conjugation, then transformation, and third is transduction. Next, if you look into binary fission is also called as amitosis because simply a single cell will divide into two. Karyokinesis, cytokinesis happens simultaneously without any cell membrane division. This is called as amitosis because there is no involvement of spindle apparatus and there is no formation of spindle fibers only. This is most common type of asexual reproduction that you can see in all the Monera. Next, endospore formation. This happens during unfavorable conditions. During unfavorable condition, bacteria produces or forms a complex coat over it, wherein covering the DNA and the core. DNA and the core, you have inner membrane. Over the inner membrane, you have germ cell wall. Over germ cell wall, you have cortex. This cortex is made up of calcium dipicoic acid, over which you have spore coat, over which you have exosporium. This much tough and thick wall is formed around the bacteria during unfavorable conditions. During what? Unfavorable conditions. So yes, the endospore has low water content, low metabolic activity, and they are unharmed during pasteurization. Endosperms endospore doesn't act as reproduction, but they act as what ma? Organs of perennation. They act as they are the only means of perennation, not reproduction. Endospores are the only means of perennation, not reproduction. Next, in favorable conditions, whenever the favorable conditions are there, then this endospore will cleave its wall and they will release new bacteria. They will cleave its wall and they will release the new bacteria. As such, one endospore can release only one bacteria. For example, there are 50 endospore formed in a colony, then how many bacteria they can release? 50 bacteria. Okay, as such, okay, one endospore releases only one bacteria. Coming to sexual recombination, in sexual recombination, there is no gamete formation or gametic fusion. There is no gamete formation or gametic fusion. Only gene recombination takes place. How? For example, in transformation though, how it happens is an experimentally proved by Griffith's experiment, which is conducted on Streptococcus pneumonia. This is conducted on what bacteria beta? Streptococcus pneumonia, wherein he has cultivated it in two culture plates, one of R strain, one type is of R strain and the other type is of S strain. Other type is of what ma? S strain. Those bacteria whichever are found in the S strain type, they are shiny because of the coat, polysaccharide. They have a coat over the bacteria that is called as polysaccharide coat, which makes the bacteria more virulent, whereas R strain bacteria doesn't have any coat over it. They are just rough bacteria, rough strain of bacteria. They are non-virulent. Keep it in your mind, wherein R strain is non-virulent, S strain is virulent. It is because they have a coat, polysaccharide coat over the bacteria. Now coming to the experiment, what is the experiment conducted by Griffith on Streptococcus pneumonia? This is, he has injected purely S strain bacteria into mice, and as the S strain bacteria can survive, it is virulent bacteria, right? So the mice is dead. Then R strain non-virulent bacteria is injected to the mice is alive. Heat killed S strain bacteria. So S strain bacteria, when it is heat killed, the coat over it, polysaccharide coat over it got degenerated, and when heat killed S strain is injected to the mice is alive. Living R strain and dead S strain, he has mixed what ma? Living R strain plus heat killed S strain. Then the mice is dead. Here he got to know that something is transforming from heat killed S strain to R strain, wherein R strain got virulent. R strain got what ma? Virulent over here, and the mice is dead. Something is being transferred from S strain to R strain is what he has explained from this experiment. Okay. Here there is no contact between donor and recipient. In the transformation, there is no contact between donor and recipient for gene recombination to happen. It is picking the DNA from the solution which is beside it or from the environment around it. Like R strain and S strain, when they are mixed, R strain has picked up the polysaccharide coats or the material from the S strain and it got transformed to virulent form as such. Okay, there is no contact between donor bacteria and recipient bacteria, but then the DNA is being taken from the environment which is around it. Okay, this is called as what? Competence. What is competence? It is asked one time. Competence is nothing but ability to pick the DNA from the solution. Ability of a bacteria to pick DNA from a solution. Here no bacteria is involved in transforming the gene or no bacteriophage is involved in transforming the gene. The DNA is just picked from the surrounding environment of the bacteria. Next, if you see conjugation. Conjugation is nothing but here you have a donor bacteria and a recipient bacteria. We have already discussed bacteria will have two types of genomes. Bacteria will have two types of genomes. One is genomic bacteria and the other is plasmid DNA. One is genomic DNA and the other is plasmid DNA. We have already discussed plasmid DNA will contain many factors like F factor, Ti plasmid. As such, they will imbibe a specific phenotypic character on a bacteria. If there is F factor over this plasmid, you will call it as F plus strain. If there is no F factor on the plasmid, you will call it as F minus strain. Conjugation is nothing but the transformation of the F factor from one plasmid DNA to the bacteria of other plasmid DNA. F plus strain and F minus strain will give you what? F plus strain of the bacteria. Okay, the daughter cell now will have the F plus strain. Here the gene recombination is happening from plasmid DNA to a plasmid DNA, from donor to recipient. F plus strain and F minus strain, finally it will give the F plus strain. Okay, so here it is F plus and F minus. The DNA is being transformed by through the process called as conjugation. In between you have pillars or pili. Okay, this is called as sex pili. What do you call it as? Sex pili or conjugation pili, which connects the two bacteria, and it will carry this F factor through this conjugation or sex pili towards the plasmid DNA of the other bacteria. Here the gene recombination happens and this also turns to F plus strain. If in case F factor is present in the genomic DNA, then it is called as HFR strain. If F factor is present in genomic DNA, what you will call it as beta? HFR strain. When it is combined with F minus strain, this also will give the F plus strain to the recipient bacteria. Clear? Is this fine? This is all about conjugation. Next is transduction. If gene recombination is happening or transfer of genetic material through a bacteriophage, is called as transduction. Transfer of genetic material through bacteriophage. Is called as transduction. Okay, bacterial DNA is moved from one bacteria to another virus and then to a bacteria. Okay, next, if you look into useful bacteria and harmful bacteria, for example, you have lactobacillus under useful bacteria wherein it makes curd from the milk. Second is antibiotics, Bacillus subtilis, the antibiotic which it is making is subtilin. Next, nitrogen fixation in leguminous roots are of two types, rhizobium and frankia, of which rhizobium is symbiotic, frankia are free living bacteria, free living nitrogen fixing bacteria attached to alnus plants. Next, harmful bacteria include cholera, which causes Vibrio cholerae, typhoid caused by Salmonella typhi, tetanus by Clostridium tetani, citrus canker in the plants by Xanthomonas citri. These are all about eubacteria. Next, coming to mycoplasma. Mycoplasma, if you see, we have discussed in the beginning only, these are what ma? These are smallest living cell. Mycoplasma is the smallest living cell and these are called as jokers of plant kingdom. It is because they don't have a specific structure. No specific structure. The structure of this organism keeps on changing with respect to the environment around that bacteria. Okay, based on the environmental conditions, based on the conditions around that bacteria, its structure is keeping on changing. The morphology of bacteria is not certain. That is why it is called as jokers of plant kingdom. And there is no cell wall of Mycoplasma. They are pleomorphic, which has many structures, many morphological structures. For example, pleuropneumonia like organisms. Next, they are facultative anaerobes. Mycoplasma are what ma? They are facultative anaerobes, means most of the condition they are aerobes. Whenever it is deprived of oxygen, then it can act as anaerobic, then it can survive in the anaerobic conditions as well. That is why it is called as facultative anaerobes. Next, mostly they are pathogenic to animals and plants. Mycoplasma are mostly pathogenic to animals and plants. If you look into the examples of Mycoplasma, it includes witch's broom, pleuropneumonia and Mycoplasma urethritis. These examples are important. You need to remember, they can be asked as match the following questions, or examples of Mycoplasma they can give and they can ask you. Okay, important for examples. This is all about the kingdom Monera. So, kingdom Monera is divided into 4 based on their presence or absence of the cell wall or the amount of cell wall. Firstly, it is Archaebacteria, then Eubacteria, Mycoplasma and Actinomycetes. For NEET though, these 3 are enough. Archaebacteria are again of 3 types based on the habitat they are living: halophils, thermoacidophils and methanogens. Under Eubacteria, the most focused topic is blue-green algae, that is cyanobacteria, and types of reproduction is again very much important. We have discussed about the experimental proofs of those sexual recombination as well. And third type is Mycoplasma, where you have to remember it is the smallest living cell. It can easily pass through the bacterial filter paper as it is the smallest living cell. They are pleomorphic, they are called as jokers of plant kingdom, and examples of this is very important. Thank you students, we will meet with another concept tomorrow.